Good evening everyone. In our last class we had started with luminometry, and we have seen that luminometry basically deals with the weak emissions of visible light which are being generated from within the samples. Okay, and due to this reason we do not need lamp and the excitation optics over here, and since these are weak emissions, we need to place the detector very close to the sample so that we do not lose any information, and this will also improve the signal to noise ratio. Okay. However, in the case of microplate luminometers, the PMT has to be kept at a distance to prevent crosstalk, and we have also talked about this because if you see the point number, the previous point, this point number 5 and point number 6, they are in contradiction, and we also talked about here that in microplate luminometers we have this fiber optic cables which take information from the microplates, and it delivers the photon signals to the detector. Okay. Then we have discussed about the crosstalk, what is a crosstalk and how it affects the signals in the adjacent realms, and how it can affect the signal to noise ratio of a given solution. Okay. Then we had briefly discussed about the instrumentation part. Okay. So in the instrumentation part, we have discussed what are the various components of a luminometer, and we have discussed over there. We had also discussed about the other important parts of a luminometer. We have discussed about the dark chambers. We have discussed about the injectors, how injectors can play an important role in understanding the kinetics of a reaction, or if there are any reactions which happens very quickly, in those cases also how we can capture the signals with the help of the injectors, right? And we had also discussed about the different types of luminometers. So we discussed about the tube luminometers, wherein we have test tubes which are being inserted into the luminometer, and the luminous or the visible light which is being generated, it is being captured. Then we had discussed about the microplate luminometers and how they function. Then we had also discussed about the multi-mode microplate readers, right? Now coming to the next topic, bioluminescence. Okay. So this bioluminescence, as the name suggests, there are two components, bio and luminescence. So bioluminescence is a process of luminescence which occurs within the biology. Okay, biology means within the biological systems. As for examples, these bioluminescence are pretty common in plankton, in algae, in jellyfish, mollusk, fireflies, firely squid, and crustaceans. Okay, the common thing in all of these is that they have a chemical compound, luciferin. Okay, they have a chemical compound luciferin, which is the chemical compound which is present within the biological system. Now this chemical compound, it is being catalyzed by the enzyme luciferase. Okay, and it acts as a catalyst for the conversion of the luciferin into oxyluciferin. Okay, and basically what it is doing, it is basically oxidizing the chemical compound luciferin. Okay, so basically this luciferase, it is acting as a catalyst for the oxidation of the luciferin compound. Okay, and the process of this oxidation, it results in the bioluminescence. Okay, and as a byproduct we get the oxidative product of the luciferin, which is regarded as oxyluciferin. So see, oxyluciferin, it is a byproduct of the bioluminescence process. Okay, herein The bioluminescence which occurs, as I told you, these are visible lights. They must be having some wavelength, and the wavelength of the bioluminescence, it is usually in the range of 440 to 79 nanometer. Okay, within this range, we usually see the bioluminescence process. Okay, this is the chemical reaction of yeah, conversion of the luciferin to oxyluciferin. So we have the luciferin over here in conjunction with ATP and oxygen. It is being oxidized into oxyluciferin. This ATP, it is being consumed and it forms inorganic pyrophosphate. Then AMP is formed and this CO2 is also formed. Okay. So during this process, there is a generation of the light. Okay. And this oxidation process, it is being catalyzed by the luciferase enzyme and magnesium ions. Okay, so if you see, the main reason for the emission of the light is that the d-luciferin, it is being converted into oxyluciferin, and in the process of this conversion we have this light which is being generated. Now in bioluminescence, there is a what you call assay, which is called Brett assay. Now this Brett assay, it is basically regarded as bioluminescence resonance energy transfer. Okay, so what happens over here is that in Brett, there is an energy transfer between one light emitting molecule, typically a luciferase, and a light sensitive molecule, typically a fluorescent protein. Okay. So herein the interaction of two bioluminescent proteins, it can be studied. Okay, and these have been used successfully in mapping neural connections within the brains. Okay, and it helps us in studying the functioning of the brains. Okay, now how does it work? It basically, if you see, there are two proteins, protein A and protein B. There are two proteins. In one of the protein, you have the luciferin compound which has been conjugated. So luciferin, it is a what do you call yeah. So it is the donor, right? So R-Luc, it is the renilla luciferase enzyme. R-Luc, it is the renilla luciferase enzyme. So it is the enzyme, and this enzyme, it is being conjugated with the protein A. Okay. Now there is another protein B. Now in this protein B, it is being conjugated with EYFP. What is EYFP? EYFP is enhanced yellow fluorescent protein. Okay. Now what happens is that if this protein A and if this protein B, they form associative interaction, that means if they are interacting with each other, the distance between this R-Luc enzyme and the EYFP protein will also be very near. And what do you mean by very near? We mean to say that the distance between these two proteins would be less than 10 nanometer. Then only we would say that this protein A and protein B, they are interacting with each other in an associative manner. Okay. Now under this condition, you will be having coelentrazarine over here. This coelentrazarine, it acts as a substrate, okay, and it is being converted into coelentramide, coelentramide, okay. Elenterazine, coelentrazine, it is being converted into coelentramide. Now, when it is being converted into the coelentramide, during that process, the R-Luke emission at 480 nanometer, it is being generated. Okay. However, if protein A and protein B, they are far apart, then these emissions cannot be taken up by the EYFP. Okay, and these lights, they are not able to excite the EYFP protein. Okay, so there will be no emission from the EYFP protein. Okay, there will be no emission from the EYFP protein. In the second instance, when the protein A and protein B, they interact and the distance between them, it is less than 10 nanometer, becomes so close that the light which is being emitted by the R-Luke coelentrazine system, it is being able to get transferred, or it is being able to excite the EYFP protein. Okay. So under that condition, what is happening? There is a secondary emission. Okay, there is a secondary emission. Now again, see, if you see that secondary emission, it is having, or it is occurring at a higher wavelength. Okay, so if you see herein, I have told that there is a fluorescent protein. EYFP, it is a fluorescent protein. Now, when I am talking about fluorescent protein, that means there is a process of fluorescence which is happening within the system. But in the case of fluorescence, we have discussed that there is a need for a source. Now, this R-Luke coelentrazine system, it is acting as a radiation source for the EYFP. Okay. However, the energy which will be transferred to the EYFP, it would be sufficient enough to induce the fluorescence in the EYFP only and only if both the systems have come to a very near position, and that distance, it is, as I already told you, it is less than 10 nanometer. Okay, so basically BRET, it is a fluorescence, this BRET, it is basically employing the fluorescence mechanism. Okay, wherein the excitation source is within the biological system. Okay, just like in the fluorescence, if you look into the wavelength and the absorption, you will see that there is an excitation maximum and there is an emission maximum. Okay, there is an excitation maximum, there is an emission maximum. Are you getting my point? And this emission, the wavelength of the emission light, it would always be greater than the wavelength of the excitation light. Okay, any questions? Hello, can you guys hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Now, where the bioluminescence is happening? The bioluminescence is happening in this area with the R-Luke enzyme, which has been conjugated with the protein A. So there are different types of BRET analysis. So if you see BRET 1, BRET 2, E-BRET 2, BRET 3, QD BRET, nano BRET. Okay. Now here the donors are R-Luke. R-Luke is this renilla luciferase enzyme. So this is the recombinant form of R-Luke8. It is the recombinant form of the renilla luciferase enzyme. Then we can have the uh, luciferase from the firefly. Then R-Luke-R-Late combination is there in QD bread, and then we have the nanolic, which is a registered trademark of a company. Okay, so substrate over here, coelenterazine for the bread 1 system. Bread 2 system, it is deep blue CTM. Then E bread 2, it is again deep blue CTM. TM, this TM stands for the trademark. Bread 3, the substrate is luciferin. QD bread is again coelentrazine, and nano bread it is furimazine. Okay. Then these are the donor emissions. Then acceptor, in the first case we use EYFP, which is the enhanced yellow fluorescent protein. Then we have for bread 2 GFP, green fluorescent protein, again green fluorescent protein. Then DS red, red fluorescent protein. Q dot, this is the quantum dot. Then halotag ligand, it is there. Now again acceptor emission 530, 510, 510, 583, 605 and 618. One important point is that when you are using the BRET 1, which is the most conventional method, to see the donor emission and the acceptor donor emission and the acceptor emission, if you see there is hardly 50 nanometer difference in the wavelength. So there are chances of, if you do not use very good quality optics, there are chances of this crosstalk. Crosstalk between the donor emission and the acceptor emission. Okay. So you have to use very high quality optics for this. However, if you see in the from BRED 2 onwards, 395, 510, there is a long gap between the wavelengths, donor emission and the acceptor emission. Similarly, in the E-BRED 2 also the same thing. Then again in the BRED 3, where we use firefly lucifer and firefly luciferase enzyme, and we use the luciferin this substrate 565 and 583 is 565 is the donor emission, and the acceptor emission is the 583. Again there is a very narrow gap between the donor emission and the acceptor emission. However, in the QD bread, if you see this wavelength separation, it is again very broad. Again in nano bread, the QD separation, the wavelength separation is quite broad. Okay, so from here the takeaway point is that when you are working with BRET 1 and BRET 3 methods, you have to be very careful with the optical systems. Okay, you have to use very high quality optics, otherwise there are chances of overlapping of the donor emission and the acceptor emissions. Okay. Now over here I have given about some of the basic information about the BRET analysis. So BRET 1, it is the original BRET method, and it is characterized by 2 strong signals and long lifetime. Even though you are having a very small wavelength separation, but in this case you can get strong signals, and there will be a long lifetime. Okay. In the case of BRET 2, this method has a better separated donor and acceptor emissions as compared to BRET 1. A better choice for screening assays where high signal to noise ratios are required. Limitation: low light emission and the short life, a short lifetime. Then enhanced breast, approximately 5-fold better signal as in the original bread 2 version. Okay. In bread 3, lower cellular autofluorescence at the emission wavelength of 565 nanometer, but the disadvantages are weak signals and overlap between donor and acceptor emission peaks. Okay. Then we have quantum dot grid. The emission peaks are clearly separated, ideal for screening applications. Disadvantages are the large size of the quantum dot molecules and the fact that the genetically coding of QD proteins is not possible. QD proteins cannot be expressed in living cells but must be added. Whereas in the previous cases, wherever the proteins are there, they can be expressed within the cells itself. Then we have nanobread. It uses luciferase enzyme, which are much brighter than the conventional luciferase enzyme. So, this luciferase enzyme is regarded as the nanolic, and it is the um, synthetically modified enzyme. It has a very good separation between the donor emission and the acceptor emission. Now, if you see, in all these cases, the basic principle mechanism, it remains the same. Now, if I give you uh, a short note to write down, say, ebred 2, then in the case of this R-Luke, you have to replace with the R-Luke 8. Then substrate, you have to use this deep blue C. Okay, and this acceptor protein you have to write as the GFP protein, and all other mechanisms over here it would remain the same. You just change these things. Okay. Then again, these emissions you need to change. From here, donor emission. What is the donor emission? What is the acceptor emission? You need to discuss about this. Okay. Just like bread, there is another analysis in fact, which is regarded as spread. It is not a bioluminescence process. And so if I ask you to write a short note or a descriptive note on bioluminescence, this topic should not be discussed over there. Okay. I am discussing it because it is similar to bread. However, it is still a separate type of analysis. Okay. Over here, FRET, it is also regarded as foster transfer. Over here, the protein which is conjugated with protein A, it has to be excited with an excitation laser or external source. Here, we need to excite the primary molecule with with an external radiation source. This is an important thing. So in bread there is no external radiation source. However, in bread there is an external radiation source. It will excite and the primary protein, the donor protein, and then it will show some emission, and this emission it is being transferred to the next protein if they are pretty close to each other. Okay, so this is the only difference between the bread and the fret. Okay. Now there is another important bioluminescence thing or analysis, which is regarded as chemiluminescence immunoassay. In this case, it is similar to ELISA. However, herein you have to you will have some chemical compounds which will give you luminescence after chemical reaction. Okay, so what happens, say for example, you have a microtiter well, and in that well you have immobilized the antibodies. Okay, then you are given an analyte within the microtiter well, and if they are specific to this antibody. These analytes are specific to this antibody. They will go and bind with the antibody. Then thereafter we will give a wash, and if there are any excess analytes, they will be wiped out. Then what happens? Then we will be adding antibody biotin complex. Antibody biotin complex. Okay, so this is the secondary antibody which is being added, and with that we have the biotin. So these biotin molecules are being attached over the analytes, and now they become immobilized over there. Then again we give some washing to wash off these excess antibody biotin complex. Okay, then we have to add avidin enzyme complex. Okay, avidin enzyme complex or conjugate. Now this over here, what happens is that this biotin, it has affinity for the avidin. Okay, biotin has an affinity for the avidin, and accordingly they will, if you see, this is the avidin enzyme conjugate. Okay, so through the avidin ligands, it will bind to the biotin molecules. Okay, and then we add a chemical luminol. Now this luminol, it is being catalyzed by this enzyme. Okay, the luminol, it is being catalyzed by this enzyme, and that gives us light. Okay, now if you see over here, what is happening, depending upon what is the concentration of the analyte, if you have smaller number of analyte over here, you will be having a lower amount of light. Okay, so however, if you have a higher number of analyte, you can have higher light intensity. Okay, so in this way you will be able to determine what is happening in the this chemiluminescence immunoassay. Okay, now the next is reporter gene assay. If you see this whole slide, you will find it that there is a lot of things over here. However, I will go to the next slide and then I will come back to this slide. Okay, now over here, what is happening is that in reporter gene assay, suppose there are two gene sequences. One is the promoter of interest and the second is gene of interest. Okay, now under the normal biological substances, under the normal biological conditions, what happens is that this promoter of interest, it will promote the gene of interest to synthesize proteins. Okay, however, this protein, it is not what you call bioluminescent in property. In such case, we want to see whether a particular condition, it can promote the activity or demote the activity of the promoter of the interest or the promoter gene. In that case, what happens is that we cut out this gene of interest, and in place of this gene of interest, we put a reporter gene. Okay, so then what we have a combination of a promoter and promoter gene and a reporter gene. Okay. Now this reporter gene, it is able to synthesize reporter protein which will generate light and which can be detected. Okay. Now what happens? This promoter of the interest if it is activated, it will be generating a large number of the reporter protein and it will give an indication, yeah, the promoter it is getting activated. If there is a reduction in the detection of the signal of the bioluminescent signal, that would mean that the activity of the promoter it has been reduced. Now if you see, as we said, the reporter gene assay, what is happening is that we are not talking about the activity of the reporter gene. In fact, we are interested in this in the activity of the promoter through this reporter gene. Reporter usually what happens, say chemical analysis. If you say chemical analysis, okay, or chemical analyze assay. So in that, what we mean, there will be some chemical reaction and all this thing. So whatever information is there, we directly relate that with that assay. However, in this case, we are telling it as a reporter gene assay, but we are not directly analyzing the activity of the reporter gene. However, we are using the reporter gene as a tool to understand the activity of the promoter gene. Okay, I hope all of you have understood. So in this case, if you see the reporter gene assay, they have been used to analyze the signaling pathways, gene regulation, and structural regulatory elements. Now these reporter gene assays, they are invaluable tool in the studies of gene expression. And over here we have a gene which consists of two main functional elements. The first consists of a so-called coding DNA sequence that provides the information about the protein being produced. Second, a specific promoter sequence linked to the coding region that regulates the transcription of the gene. Now, this promoter, what it said, this promoter gene, it is affecting the activity of the gene of interest to produce or synthesize the protein. Over here, definitely, this protein will not be generated, but it can allow you to understand under what condition this gene of interest may be activated or it may be its activity would be hindered. Okay. The main purpose of the reporter gene assay is to investigate the promoter of a gene of interest, that is the regulation of its expression. You're getting my point? This can be done by linking the promoter of interest to an easily detectable gene such as the gene of firefly luciferase, which catalyzes a reaction that produces light. Usually the cells are then exposed to different factors or conditions, or changes can be made in the orbit of the reporter, the effect of which can be easily tracked by measuring the changes in the light emission. Okay. So what happens? Many of you must have heard about the plasmids. So what you do in the plasmids, there are two areas: the promoter area, then this gene of interest. I will mark the gene of interest as black. So using the restriction enzyme, we will take out this gene of interest, and instead of that, say we will put, it is too dark. Now we will put in this blue colored system, which is the reporter gene, RP. Okay. So this was the initial case. Okay, this was the initial case. The first case is the initial case. So what we have done in the second stage, we have removed the gene of interest and we have replaced with the reporter gene. Now these plasmids, they are usually transfected within the various microbial cells. One of the common cell where we studied this kind of activity is the E. coli cells. We transfect it over there, okay, and then we put the cell under different conditions, or and due to the change in the conditions, we see whether the light which is coming from the E. coli cells, they are higher or lower. So if there is a high activity, that means the promoter gene, it is being activated, and if the light is reduced, then that means the promoter gene, it is being suppressed. Okay. Then we have dual luciferase reporter assay system. In the dual luciferase reporter assay system, what happens is that we have a control reporter and we have an experimental reporter. Control means we know how the activity will change or not change. Okay. So why this is so? This is because what happens over here is when we use a combination of a control reporter and an experimental reporter, over here it allows us to get over the variability in the results. Okay, because herein we are having a biological system, E. coli, which itself is a variable stuff. So there is a complex biological processes which are going on within the biological system itself, right? So there will be some variations over there also. Now, due to this reason, what happens is that we what we do is that we include a control reporter and the experimental reporter, and usually the experimental is to the control reporter. I am writing in CR. So ER is to CR. It is either uh, given in the ratio 2:1 or 1:1. Okay. So it is either in the 2:1 format or ratio. You need to put it in within the cells, or in a 1:1 format. So from there you will be able to compare the intensities of the both the control and the experimental reporter, and from there you can do the analysis. So using this dual luciferase reporter system assay, it allows you to overcome the variability of the biological systems. Okay. However, you need to keep in mind that the emission wavelength of both these system, the control reporter and the experimental reporter, should be wide apart, so that you can analyze the data in a meaningful way. Then it can help us in detecting, in measuring the ATP content within a system. So ATP determination, it provides us information about the viable cell numbers. Okay, higher the number of cells, you will be having higher the ATP. Now, if you go over there, in the first case, in this equation, we have D-luciferin, we have ATP, we have oxygen. Okay, So oxygen is for the oxidation. The thing is that in this case, in the normal cases, what is happening? We are looking into the deluciferin, and we are assuming that ATP and oxygen and other parameters, they are in excess. Okay. However, when we try to do ATP measurement, herein we have to keep the luciferin and the other reaction products as in an excess amount, and the reaction mechanism, it will be what you call, it will be governed by the availability of higher amount of light. If ATP is low, you will be getting lower amount of ATP. Okay. Now, if you see, this reaction is very popular in where ATP and luciferin are in excess, and the quantity of light produced is limited by the expression of the luciferase enzyme in the conventional one. However, the same reaction can be used to quantify ATP if both luciferin and luciferase enzyme, they are in excess. Okay. So, as I told you, you have luciferin, D-luciferin, plus you have the ATP, plus you have the enzyme. Okay. So, D-luciferin was the constraint in the previous cases. However, in this case ATP measurement, we have to keep the D-luciferin and the enzyme, the luciferase enzyme in excess, and only the light generation will be dependent on the availability of the ATP. If there is a higher amount of ATP available, there will be a higher amount of light which will be generated, and vice versa. Other applications, we have hygiene monitoring. ATP is present in all living cells and is therefore an indicator of the biological contamination. Okay, so you can look into the system if you want a very sterile system, and if you see that some bioluminescence is being generated, then you can go for that. We have already discussed the immunoassay in the CLIA format, CLIA. Okay, then we have caspase assay. So basically we try to understand the caspase 3 activity of the caspase 3 enzyme. This caspase 3 enzyme, it is basically important in the case of apoptosis research. Okay, so we can look into this apoptosis research. Again, over here you go for the measurement of the ATP. Higher the ATP, higher will be the light generation, and that means the cells are being killed. Okay, if the caspase 3 is being activated, the mammalian cells will be dying, and if they are dying, the quantum of ATP would be coming down. If the quantum of ATP is coming down, then there will be a reduction in the light component. Okay, so what you do over here is that, suppose you have some cells, mammalian cells in petri plates or some wells. You have this media over there, then you add some agent, say analyte A, for which you want to study the anti-cancer activity. You put it over here, and after this addition, if the caspase 3 is activated, it will either kill the cells or it will have no effect on the cells. If it is killing the cells, then the ATP is coming down, and if the ATP is coming down, that means the light would also coming down. So from there you can find out the anti-cancer activity of the different drugs or chemicals. Then we have kinase assays. Kinases are modifying the activity of specific proteins and are extensively used to transmit signals and control complex processes in the cells. Their enormous diversity and their role in signal transduction make them attractive targets for research and drug design. Okay. Then cellular luminescence. Cellular chemiluminescence is the production of light after chemical reaction of activated cells like neutrophil granulocytes, monocytes, or macrophages. When phagocytosis is activated as the first immune response in order to protect the organism from invaders, after ingestion of the external object, reactive oxygen species such as H2O2, OH minus O2 negative are produced, resulting in luminescence of a very low light, a very low intensity. To enhance the chemiluminescent effect, substrates like luminol and lucigenin can be added. Then water toxicity testing. Over here we use luminous photobacteria. Examples are Vibrio fischeri and Photobacterium leognathi. Okay, or you can also use dinoflagellates for toxicity assessment. If the water is toxic and if you put in these microbes, suppose there is a water, I have given you a sample of water, then you add these microbes. Now after the addition of the microbes, they will be there. Now over a period of time, since they are photobacteria or these microbes, they are emitting the light, you will be getting some intensity of light. However, after adding it to this water, for which you want to test the toxicity, if they are toxic to the cells, this bacteria or the microbes, then what happens? It will be killing the bacteria, and if they kill the bacteria, the intensity of the light would come down over a period of time. The intensity of its luminescence is strongly affected by a wide range of contaminants, including pesticides, herbicides, chlorinated hydrocarbons, heavy metals, agents that affect the cell's integrity, membrane function, or cell respiration, or that have an impact on the rate of protein or lipid synthesis. Okay, in different way it can affect the microbes. Okay, so in this way we can find out whether a given water it is toxic or not. With this I complete the chapter of luminometer. If you have any questions, we can discuss. Do you have any question? No? Okay, then we will stop over here. Thank you very much.